Next topic, mitochondrial genome and yeast peptide mutants, which comes under the topic of organelle genome. Organelle are mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, etc. The genome present in the chloroplast and mitochondria are called as organelle genome. 5 roles mitochondria play in cells. Mitochondria are referred to as the powerhouse of cells. They play a vital role in converting food energy into energy for biological processes. Mitochondria also play a critical role in activities that enable cells to function and help to maintain a healthy body. Mitochondria are referred to as powerhouse of cells. They play a vital role in converting food energy into energy for biological processes. Mitochondria also play a critical role in activities that enable cells to function and help to maintain a healthy body. So, production of ATP, regulation of innate immunity, then calcium homeostasis, programmed cell death, stem cell regulation. Production of ATP, regulation of innate immunity, calcium homeostasis, programmed cell death, and stem cell regulation. Mitochondrial genome. This genome consists of circular chromosome, 16.5 kilobase pairs in size, that is located inside the mitochondrial organelle, not in the nucleus. Most cells containing at least 1000 empty DNA molecules, mitochondrial DNA molecule, distributed among hundreds of individual mitochondria. Not all the DNA and protein synthesized in a cell are enclosed in the DNA of the nucleus. It contains 37 genes and encodes 2 types of rRNA and 22 tRNAs. Genes encode 13 proteins that are subunits of enzymes of oxidative phosphorylation. The remaining 74 polypeptides of the oxidative phosphorylation complex are encoded by the nuclear genome. Then nuclear DNA versus mitochondrial DNA. Once again, this previously I am going to read this. Mitochondrial genome. This genome consists of circular chromosome that is located inside the mitochondrial organelle, not in the nucleus. Most cells contain at least 1000 empty DNA molecules distributed among hundreds of individual mitochondria. Not all the DNA and the RNA and protein synthesized in a cell are enclosed in the DNA of the nucleus. It contains 37 genes and encode 2 types of rRNA and 23 tRNA. Genes encode 13 proteins that are subunits of enzymes of oxidative phosphorylation. The remaining 74 polypeptides of the oxidative phosphorylation complex are encoded by the nuclear genome. Nuclear DNA versus mitochondrial DNA. Nuclear genome versus mitochondrial genome. Found in nucleus of the cell, 2 sets of 23 chromosomes, maternal and paternal, can discriminate between individuals of the same maternal lineage. Double helix bounded by a nuclear envelope, DNA packed into chromatin. But mitochondrial genome found in mitochondria of the cell, each mitochondria may have several copies of the single empty DNA molecule, maternal only, cannot discriminate between individuals of the same maternal lineage. Circular, free of a nuclear envelope, DNA is not packed into chromatin. Found in the mitochondria of the cell, each mitochondria may have several copies of the single empty DNA molecule. Maternal only, cannot discriminate between individuals of the same maternal lineage. Then circular, free of a nuclear envelope, DNA is not packed into chromatin. Then once again, nuclear DNA location, nuclear size 3200 MB, structure antiparallel double helix, linear, intron present, non-coding sequence, many non-coding sequences, about 98%. Transcription monogenic, copy number 1 set per cell. Then number per cell 46, associate proteins, histone and non-histone, encoding genes 30,000. Transcription, individual gene transcription, codon, universal codon. But empty DNA, mitochondrial DNA, location mitochondrial matrix, size 16.6 kb, Structure double strand, circular. Intron absent. Non-coding sequence, few non-coding sequences. Transcription multigenic. Copy number hundreds to thousand per cell. Number per cell several thousand. Associated proteins largely protein free. And encoding genes 37. Transcription bulk transcription for whole strand. Codon mitochondrial codon. Its own codon, mitochondrial codon. Genetic code, mitochondrial code, universal code. Characteristics is inherited exclusively from the mother. mtDNA is a circular shaped single chromosome. It is only 16 kb in length, contain 16,000 base pair, codes for 37 genes. It is only 16 kilobase pairs in length, contains 16,000 base pair, codes for 37 genes, contains 22 tRNA and 2 rRNA coding genes, encodes 13 proteins that are subunits of oxidative phosphorylation, contains only exons, no introns, has no reparation system, high mutation rate, especially in D loop, no crossing over. Mitochondria DNA structure. Mitochondria harbor a small but essential component of an eukaryotic genetic material. Animal mtDNA are extremely compact, being less than 20 kilobases in length and encode fewer than 40 genes. All of the proteins encoded by the mtDNA are subunits of the mitochondrial respiratory chain. Despite their small number, the genes on the mtDNA necessitate a large expenditure of cellular resources because they are essential for strict aerobes. mtDNA is 13,794 nucleotides in length and encode 36 genes, 2 ribosomal RNA, 12 as rRNA and 16 as rRNA, 22 transfer RNAs and 12 MRC subunits. Mitochondrial DNA structure. A typical animal mtDNA, the nematode mitochondrial genome is slightly smaller than its human counterpart. It lacks the ATP8 gene of the human genome, which encodes a subunit of ATP synthase complex 5. Introns are absent and there are few or no non-coding nucleotides between genes. Approximately 92% of the mitochondrial genome has coding function. 3 genes end with incomplete termination codons, T or TA, that are completely converted to TAA codons by polyadenylation of the transcript. Only one sizable non-coding region called the displacement loop or D loop is present. The molecule containing the genes for 12 protein, thick grey arrows shown here. The 2 rRNAs, black arrows, and 22 tRNA, circles labeled with one letter amino acid code. The serine and leucine tRNAs are also identified by the codon family organized. The positions of the purate D loop and the UA DF5 deletion mutation are indicated inside and outside the circle respectively. Cl against mtDNA. The maternal inheritance of mtDNA. During fertilization, the sperm only contributes its nucleus, 23 chromosomes. Mitochondria of sperm cells are located at the mitochondrial sheath, which is destroyed upon fertilization. Only available mitochondria, mtDNA, is that of the mother. That is why mtDNA is of maternal origin. Nuclear DNA inherited from all ancestors. Mitochondrial DNA or mtDNA inherited from a maternal lineage. The mitochondrial genome, 2 general regions. Coding region, responsible for the production of various biological molecules involved in cellular respiration. Control region, responsible for the regulation of the mtDNA molecule. It involves faster than nuclear DNA. Probably due to insufficient replication repair, mitochondrial DNA is maternally inherited in most species. Generally, mitochondrial DNA does not recombine, though some evidence for recombination events has recently been reported. Yeast mitochondrial genome. The mitochondrial genome of yeast is large. its genes contain introns. It contains genes for the tRNAs for the respiratory chain, cytochrome oxidase 1, 2, and 3, cytochrome B for 15S and 21S rRNAs, and for subunits 6, 8, and 9 of the ATPase system. The yeast mitochondrial genome is remarkable because its ribosomal RNA genes are separated. The genes for 21S rRNA contain an intron. About 25% of mitochondrial genome of yeast contains AT-rich DNA without a coding function. The genetic code of the mitochondrial genome differs from the universal code in nuclear DNA with respect to usage of some codons. Mitochondrial genes in yeast. Next, mitochondrial inheritance. Yeast has been used extensively to study mitochondrial inheritance. There is a yeast strain called peptide that have structurally abnormal mitochondria that are incapable of oxidative phosphorylation. These mitochondria have lost some or all of their DNA. Genetic crosses between peptide and WT strains showed that inheritance of this trait did not segregate with any of the nuclear chromosomes. So, there is a yeast strain called peptide that have structurally abnormal mitochondria that are incapable of oxidative phosphorylation. These mitochondria have lost some or all of their DNA. Genetic crosses between peptide and WT strains showed that inheritance of this trait did not segregate with any of the nuclear chromosomes. Peptide mutation. Peptide is a mutant first discovered in the yeast Saccharomyces cerevisiae due to the defect in the respiratory chain. Peptide yeast are unable to grow on media containing only non-fermentable carbon sources such as glycerol or ethanol. Peptide is a mutant first discovered in the yeast Saccharomyces cerevisiae. Due to the defect in the respiratory chain, peptide yeast are unable to grow on media containing only non-fermentable carbon sources such as glycerol or ethanol, and form small colonies when grown in the presence of fermentable carbon sources such as glucose. Peptide is a mutant first discovered in the yeast Saccharomyces cerevisiae. Due to the defect in the respiratory chain, peptide yeast are unable to grow on media containing only non-fermentable carbon sources such as glycerol or ethanol, and form small colonies when grown in the presence of fermentable carbon sources such as glucose. The peptide phenotype can be caused by the absence or mutations in mitochondrial DNA termed cytoplasmic peptides, or on mutations in nuclear encoded genes involved in oxidative phosphorylation. A neutral peptide produces all wild type progeny when crossed with wild type. Peptide mutations can be induced using a variety of mutants, including DNA intercalating agents, as well as chemicals that can interfere with DNA synthesis in growing cells. Mutagens that create peptides are implicated in decreased rates of degenerative diseases and the aging process. Mutagens that create peptides are implicated in increased rates of degenerative diseases and in the aging process. Causes. The peptide is characterized by a deficiency in cytochrome A3 plus B and a lack of respiratory enzymes which engage in respiration in mitochondria. Due to the error in the respiratory chain pathway, peptide yeast is incapable of growing on media containing only non-fermentable carbon sources such as glycerol or ethanol, and forming small colonies when grown in the presence of fermentable carbon sources such as glucose. The absence of mitochondria can cause the peptide phenotype, or by deletion mutations in mitochondrial DNA termed cytoplasmic peptides, which is a deletion mutation, or by mutations in nuclear encoded genes involved in oxidative phosphorylation. Peptide mutants can be generated in the laboratory by using high efficiency treatments such as acryflavine, ethidium bromide, and other intercalating agents. Their mechanism work to break down and cause the eventual loss of mitochondrial DNA. If the treatment time increases, the amount of mitochondrial DNA will decrease. After prolonged treatment, peptides containing no defectable mitochondrial DNA were obtained. It is a useful approach to illustrate the function of mitochondrial DNA in yeast growth. Yeast peptide mutants. Yeast can grow either anaerobically by fermentation, slow growth, or aerobically using mitochondria. Fast growth, forming colonies from single cells on solid media. Yeast peptide colonies are much smaller than those formed by wild type cells due to cytochrome deficiencies that prevent aerobic respiration. On a medium that supports only aerobic respiration, peptide cells are unable to grow. The spontaneous mutation rate is 0.1 to 1%, but exposure to an intercalating agent, ethidium bromide, raises the rate to 100%. This allows isolation of different peptide cell lines containing different mutations. Yeast crosses between peptide and wild type cells determine the mechanism of inheritance for this phenotype. The zygote formed from mating is grown into a colony to check its phenotype, and when it sporulates by meiosis, the tetrad of ascospores can also be grown into colonies for phenotype analysis. Some peptide X wild type crosses give 2 raised to 2 segregation wild type peptide. This is the same ratio as seen in nuclear gene, so these peptide mutants are nuclear segregational peptides, written PET. The cross in this case was PET X PET plus, diploid was PET plus negative and plus PET positive, hence wild type, and the spores tetrad contained 2 PET negative and 2 PET positive spores. Mitochondrial inheritance in yeast, normal mitochondrion, haploid placenta with wild type nuclear genes, peptide mitochondrion, mating by cell fusion, diploid zygote, meiosis, random distribution of mitochondria to daughter cells, all haploid cells respiratory proficient. Mitosis, random distribution of mitochondria to daughter cells. Mitosis, meiosis, respiratory proficient peptide, respiratory proficient. Peptide mutation inheritance. The inheritance pattern of genes existing in the cell organelles such as mitochondria, which named cytoplasmic inheritance, differ from nuclear gene pattern. Peptide mutation inheritance types. Peptide mutants show extranuclear inheritance. The inheritance pattern varying with the type of peptide involved. Segregational peptides, PET negative mutants, are created by nuclear mutations and exhibit Mendelian 1+1 segregation. Neutral peptides, rho N. Neutral peptides, when crossed to wild type, all offsprings are wild type. It has inherited normal mitochondrial DNA from wild type parent, which is replicated in the offspring. Then suppressive peptides, rho S, crosses between peptide and wild type. All offsprings are peptide, showing dominant behavior to suppress wild type mitochondrial function. Then peptide mutant crosses, segregational, haploid peptide, haploid normal. Then diploid zygote normal, sporulation, meiosis, haploid ascospores, peptides are normal. Then neutral, segregational peptide crosses. Haploid peptide, haploid normal, diploid zygote normal, sporulation, meiosis, haploid ascospores, peptide and normal. Neutral, haploid peptide, haploid normal, diploid zygote normal, sporulation, meiosis, haploid ascospores, all normal. Then suppressive, haploid peptide, haploid normal, diploid zygote, usually peptide, sporulation and meiosis, haploid ascospores, all peptide. Thank you for listening.